I'm Julia, and here is our Enzyme Lab. So, starting off, trypsin is a proteolytic enzyme that is important to the digestion of proteins. So it cleaves the terminal hexapeptide to form a single chain called beta trypsin. The two primary forms of trypsin are alpha trypsin and beta trypsin. Trypsin degrades proteins, and the optimum pH of trypsin is 8 in the small intestine of humans. And so at this pH, it reaches its maximum enzyme activity. So our question was, would pH of 8 be the optimum for enzyme activity for trypsin? And our hypothesis would be if we were to lower the pH to a more acidic buffer, then the rate of enzyme activity would decrease. And our rationale for this was because we knew that the pH of 8 is the natural pH in the small intestines, so changing it would lower enzyme activity. For our materials and methods, so our steps. So first off, we added 450 microliters of each buffer. So between all of the buffers, they had the same salt concentration, so there wouldn't be any variability between, nothing to take into account of when we're measuring. So we did a pH of 5 through 11, and we did one negative control with distilled water replacing our trypsin. So then after we added all the pHs, we added 500 microliters of azocasein to each tube. And then after that, we added our 50 microliters of trypsin. So then we incubated at 35.8 degrees Celsius for 20 minutes to regulate human body temperature as close as possible. And then we added 500 microliters of trichloroacetic acid, which is called TCA. So we added TCA because it's a proteolytic enzyme that also hydrolyzes peptide bonds and shorter bonds, and amino acids are liberated from the chain. Trichloroacetic acid is added to stop the enzyme basically from working. So then after we added our TCA, we centrifuged the test tubes at maximum speed, which was 14,000 RPM, for 2 minutes. And then after that, we transferred the supernatant to a clean cuvette. We blanked the spectrophotometer to get a calibrated good reading. And then we read the absorbance in the spectrophotometer at 440 nanometers. So why do we centrifuge the tubes? We centrifuge the reaction so then it can separate the precipitated macromolecules, including the enzyme and undigested and azocasein. So they're both removed from the reaction by centrifuging it. So very short peptide chains and free amino acids are liberated, but are not precipitated by TCA, and thus they remain in solution. So what was our substrate being used? So trypsin is a pancreatic serine protease with substrate specifically based upon positively charged lysine and arginine side chains. The enzyme is excreted by the pancreas, and it takes part in the digestion of food proteins and other biological processes. And then sulfanilamide allows for the absorbance to occur at the 440 nanometers at the spectrophotometer. Now for our results. Trypsin enzyme activity amongst varying pH values is not greater than enzyme activity in the negative control containing the distilled water rather than trypsin, meaning that there is a 59% chance that these results were due to chance. So the pH of 10 appears to be our optimum, but this conclusion is not reliable because of a substantially large outlier of 1.094 amongst the trials for pH of 10. So there's approximately a 20% increase from the averages of 6, which is here, to 10, as you can see. Then from pHs 10 to 11, there is a percent decrease approximately of 30%. So seeing it from 10 to 11, it's a decrease. Further suggesting that it's likely that 10 is the optimal pH value, but variability amongst the trials was substantial, but still relatively low, ranging from 9% to 37%. So our smallest p-value, which was 4.42 times 10 to the negative 6, and then with the highest level of certainty was at pH of 8. So our graph represents trypsin enzyme activity versus varying pH values, but our enzyme activity numbers are all averages. So the average rates of trypsin enzyme activity from the range of 5 to 11. So the statistical analysis was conducted through the use of paired sample t-tests. So the level of activity at each pH value is then compared to a negative control containing our distilled water, which is pictured here, which is obviously the lowest. So we used a salt concentration of 0.15 molar and a pH value of 8 for our negative control at constant temperature at 35.8 degrees Celsius, which is closest to our human temperature. So throughout the experiment, enzyme activity at pH 10 is significantly higher than what we thought it would be at. And so, within the exception of 9, which is higher than 8, so it goes. So 8 is proven to not be our highest enzyme activity level, but we can't quite rule that out yet because we did have the outlier in 10. And on this graph, you can see the small bars here. That is our standard deviation, and as you can see, at pH of 10, the standard deviation is much higher than 8, so that's another reason why we can't completely trust our results. So now for our discussion. So our experimental design, we used a pH ranging from 5 to 11. We centrifuged the tubes that were incubated for 20 minutes at 35.8 degrees Celsius. And our negative control was distilled water in place of trypsin, but the same volume with the same buffer of pH of 8. So, but was it adequate? Well, so our experiment itself was adequate, but it did not fully satisfy our hypothesis based on our data, as we talked previously about. So the data did not support the hypothesis because there's too much variability within the results, and a replicate with the pH of 10 had an outlier in it, which is why it's not conclusive that these are fully 100% reliable results. For our controls, we used different levels of pH ranging from 5 to 11. The similar salt concentration between all the microcentrifuge tubes, which is 0.15 molar, and our temperature at 35.8 degrees Celsius, and equal volumes of trypsin throughout to decrease variability so we can be able to adequately measure our data. So why did we choose this? So we felt that the range of pH that we chose would give enough data to be reliable. So it also reduced the measurement of variability and human error as well. Keeping equal volumes of each product will most likely ensure that we have precise data, so there's no human error coming into it. So then for our negative control, we replaced the pH buffer solution with the same volume of distilled water, keeping the enzyme and still adding the TCA with it that we mentioned earlier. So this allowed for a reading with no enzyme activity because there was no enzyme in the solution to begin with. So one of our groups in our class tested salt concentration as their independent variable, while we chose pH for ours, and they concluded that using anything greater than 0.15 molar of salt concentration will result in denaturation of the enzyme, which is why it was better to use the 0.15 as a way to keep everything normal and to minimize the effect of denaturing the enzyme. So for our replicants, we created 3 trials for every pH tested from 5 to 11. And we did this so we can increase specificity and decrease the measure of variability within our results. So for our negative control, we only used 1 replicate because we were certain there would be no reading from the tube. So we made sure that we had enough data to be able to say whether or not our data is correct, reliable, and we can make future measurements from this. If I were to redo this experiment again, I would decrease the amount of human error caused due to this outlier. So we would make sure that we pipette the correct amounts and make sure human error just as limited as possible, being as careful as we can be. And then here are our references used for this whole project. I'm Joey Vitola, and that was our Enzyme Lab.